Thank you, Dr. Wang, for the very informative presentation. Our final presenter is Dr. Roman Togowatsky. Dr. Togowatsky helps biotech CEOs, CMOs, and investors reduce drug development failure risk by uncovering and governing hidden assumptions about disease mechanism, biomarkers, endpoints, patient selection, and causal inference before those assumptions derail even technically rigorous programs. His work centers on structural sources of causal and evidentiary fragility, and this failure arises from conventional misunderstandings of how disease mechanism, physiological regulation, and systemic biological context shape symptoms and outcomes. And from gaps in causal inference methods, by addressing those blind spots, he strengthens the logic underlying high-stakes clinical development choices. He has supported programs across multiple therapeutic areas in the development stage, informing go-no-go decisions, endpoint strategies, and investment diligence, and increased confidence in high-cost, irreversible decisions. He holds a Ph.D. in biostatistics from Harvard University. Today, his presentation is entitled The Hidden Disease of Evidence Generation, Diagnosing Methodological Failure in Clinical Trials and Real-World Evidence. Those.
Thank you. So the core idea I want to share today is fairly simple, but I believe it has fairly profound impact on both drug development and the clinical medicine that we encounter. Throughout the clinical development and real-life clinical encounters, we make a number of assumptions, and these assumptions span biology, causal inference, statistics, patient-reported outcomes. And what I mean by these hidden assumptions is that we are not really aware of these assumptions, and therefore they are quite dangerous in the sense that they increase the risk of clinical development failure and regulatory pushback, which ultimately leads to financial loss. And if you take one idea that I will try to illustrate, that what I encounter constantly in my consulting practice is that we are operating on the surface, physicians, biostatisticians, we have certain belief systems, and we are not aware of these deeper, oftentimes ontological assumptions that we are making. And oftentimes we are walking on a minefield without realizing that these assumptions can explode at some point. And I'll give you a couple of illustrations. One of the core concepts in medicine and drug development is a disease mechanism. So we usually envision it as some molecular pathway that leads to certain adverse health outcomes as well as unpleasant symptoms. And we design drugs in order to target these disease mechanisms with the hope that they will improve health outcomes and reduce symptoms. What I'll try to show is that this very intuitive model is actually not very valid. Our entire notion of medicine is based on so-called biomedical reductionist assumption. We kind of envision human organism as a number of modules. These modules have stable function throughout the person's life, and they're very similar across people, and they're connected. And that's why in medicine we have different specialists specializing on different systems. Now, when something happens in the organism, we implicitly assume that it's a breakdown in a particular module. We can find that module, isolate that module, almost like what we do when we bring our broken car to a mechanic. We find the broken part, we extract it, we repair it, and we put it back. If you think about a patient who gets diagnosed at the same time with depression, sleep disorder, hypertension, and he gets prescribed one drug for each disease, that's basically an operational reality of how we function in medicine. We assume that different modules break down and we treat them with different drugs. If you look at modern science, then modern science says that this idea is actually very far from being real. We know that disease mechanism evolution is affected to a great degree by the state of autonomic nervous system, by circadian system, by sleep architecture. And if you imagine having two patients, with one patient with, for example, having no deep sleep, and another patient has beautiful deep sleep and normal function of autonomic nervous system, in real life, no matter what disease they're suffering from, these are dramatically different patients. But in the current system, we are treating these patients as absolutely the same kind of patients generated from the same distribution. And as a result, we're basically averaging over very important differences. Just to give you a real-life example, natural killer cells are quite important in oncology. They're part of the immune system, so the natural assumption is that, well, they're regulated by the immune system. Now, the reality is that they're regulated by deep sleep, by sympathetic dominance. So all of these factors actually suppress the cytotoxicity of natural killer cells. And once again, if you imagine two patients diagnosed with the same disease but having different physiological context here, these are, in real life, these are very different patients. And when we don't take that into account in clinical medicine or in drug trials, when we design them, we're basically averaging over very important contributions. We are putting all of these factors into noise. Another fundamental assumption that we are making is the notion of the control system, which is absolutely central to human disease and health. The standard notion in medical physiology, and physicians are still being taught that, is that the core task of the organism is to maintain homeostasis, and the way the organism dies so is through so-called negative feedback loops. So here you can see a body with red circular arrows which signify a bunch of negative feedback loops. Now, what happens in reality, at least what we currently know, is that the core goal of the organism is not to maintain homeostasis. It is to actually maintain function under external and internal demands. And this one idea can change real-life clinical medicine rather dramatically. The way the organism tries to maintain function under demands is by oftentimes shutting down some of the negative feedback loops. So it's not about negative loops, it's about regulating, basically operating characteristics of these negative feedback loops. And now if you imagine that you have two patients who have very different external demands or internal demands in terms of comorbidities, then most likely all the dynamic regulatory strategies for these two people are very different. But we treat them as having the same disease, and therefore we average over very important factors, which are not just treatment effect modifiers, they're actually parts of the mechanism which we erase by averaging. So just to summarize the key idea, I presented you with this very intuitive idea of drug affecting disease mechanism and ultimately affecting the outcome and patient-reported outcomes, and illustrated some of the hidden assumptions that are really never explicated, neither in drug development nor in clinical medicine. The reality is that oftentimes it's very difficult to separate a clean disease mechanism from the physiological context or from the holodynamic regulation. And in some diseases, the disease itself is just a regulatory strategy to maintain function, which is really not, unfortunately, appreciated currently in clinical medicine, but is fairly well described in the research literature. Furthermore, we kind of assume that a drug acts on the disease mechanism, and we sort of expect that the treatment effects are pretty homogeneous. Yes, we develop biomarkers, we try to find a little bit of heterogeneous effect, but the idea is still that it's more or less homogeneous. When you look at the more realistic scientific picture of how human organism works, then we realize that drugs actually may affect not only the disease mechanism, but they can also affect the physiological context and the holodynamic regulatory strategies. And this stuff oftentimes gets pushed into side effects. Now, what is even more important, that if the drug affects physiological context, then context can potentially impact the pathway from the drug to the disease mechanism. And that means that we, theoretically at least, should expect that the drug treatment effects should be context-dependent, time-varying, and heterogeneous, just simply from understanding biology, not from a statistical perspective. And that has direct implications for design of clinical trials. We should start thinking about the what control system is, how it functions, and about the physiological context. It's not realistic to believe that this idealized disease mechanism actually exists outside of the greater physiological context, maybe aside from ultra-rare some of the genetic diseases. If we actually want to help more patients, then logically we also need to look not only at the actual molecular disease mechanism, but also at the physiological context and how to develop interventions that actually impact that physiological context and regulatory strategies. And this is not just some theory which has no implications for drug development or trials. I have seen firsthand how powerful these interventions can be when they're combined with drugs that target particular disease mechanisms. Another puzzle in medicine oftentimes is we develop effective drugs that go through the trials, we improve the health outcomes and endpoints, but for some reason the symptoms or patient-reported outcomes are not moving. And that's quite puzzling because implicitly medicine believes that symptoms are generated by disease mechanisms. And there is a similar problem in so-called medically unexplained symptoms. So for those who are not familiar with this term, it's a highly prevalent set of conditions where a patient goes to see a physician, complains of certain symptoms. Physician runs a battery of tests. All the tests are fine, but the patient is suffering tremendously, as oftentimes suicidal. And our current medical system, unfortunately, fails tremendously for these patients. These patients suffer for years and sometimes decades. And all of these puzzles can be explained by the underlying hidden model that we assume how human organism works. There is a certain peripheral disease process which changes physiology. This is picked up by peripheral receptors which send information to the central nervous system, which through some magic creates experienced subjective state or symptoms. So in this model, the symptoms are more or less a passive readout of whatever disease process or whatever pathology is going on in the periphery. And naturally, if you imagine yourself being a physician, imagine seeing a patient who is suffering tremendously, experientially, but then has no identifiable disease process. Well, that's very, very puzzling, to put it mildly. Now, the reality is that human organism, from what we know now, works in a very different way. The brain cannot measure physiological state. It can only infer hidden physiological state by solving so-called inverse ill-posed problem. It's an inverse ill-posed problem because many different hidden physiological states can actually generate the same sensory signaling that is sent to the brain. So in applied math and physics, it's called ill-posed problem, and the main task of the brain is to infer or guess what is actually going on inside of our body. So again, it's not a measurement like we measure insulin. It's a statistical inference. Now, once the brain makes that inference, it actually encodes that inference, results of that inference, as subjective experience, which basically explains how different psychiatric symptoms or patient-reported outcomes are generated. Again, they're not measurements, they're results of inference. And the way the brain infers this hidden physiological state is not only through the sensory signal, which is here you can see on the left, which reflect essentially disease mechanism, but also through the generative model and physiological context, which explains why symptoms are not just passive readouts of the underlying physiology. And this also explains the puzzles. You can improve pathological process, but see no change in symptoms. Or you may have no change in pathology, but you can have improvement in symptoms simply because the generative model has changed. That has direct implications also for making sure that we're not failing at the stage of patient-reported outcomes. We need to think about the entire model, that it's not only drug that affects disease mechanism and that way it affects the symptoms, but it's also the physiological context and the generative model that determine the symptom. Now, a couple of words about similar hidden assumptions in randomized clinical trials and causal inference. In my consulting, I oftentimes kind of hear this implicit belief that we're doing randomized clinical trials, we're getting paid for doing randomized clinical trials. And I do not believe that that is our objective. We are not getting paid to run randomized clinical trials. We're getting paid for generating evidence for causal claims, because every time we say that we think that we need to prescribe certain intervention to a particular population, that's an intervention. That's the paradigm of causal inference. But ironically, from what I see, the deep expertise in causal inference in the industry is very, very thin. We're still kind of 20, 30 years ago when we were trying to run RCTs using standard statistical method, but the randomized trials are only generating the data. It's the causal inference that actually turns the data into the actual evidence that will be accepted by the regulators. A couple of examples. It's very common to use Cox hazard ratio in survival. But if you look at it, it's been known for quite a long time that actually it's not estimating any marginal causal effect. There are tons of issues in conduct and reporting of adaptive trials. If you look at the whole controversy about surrogate endpoints, that whole controversy can be explained by belief that surrogates are supposed to be associated with the endpoint of interest, but it's a purely causal statement. The surrogacy statement is a causal statement that can be displayed using DAGs. When you look at observational studies, it's actually kind of very depressing. So one of the key methodologies to build robust designs for observational studies is target trial emulation. And even out of those papers who claim to do target trial emulation, only 18% of studies actually do it properly. But that's even not the worst news. In comparative effectiveness studies, only 3% try to build a robust design for observational study, for example, by doing target trial. And if you look at externally controlled trials, that almost no externally controlled trials actually follow the basic standards of causal inference. And that basically reflects, unfortunately, a very deep structural governance failure. So it's not about one statistician not doing something. It's about the entire industry kind of falling behind the developments in causal inference. Even when companies are at the stage of trying to get reimbursed and doing population-adjusted indirect comparisons, only 1% of published papers actually do everything methodologically correctly. Although if you look at it, the methodology is available out there. Another very common deep issue is so-called Schrödinger causal inference. Basically, here the idea is that people try to do causal inference without doing causal inference, or meaning doing causal inference incorrectly. The idea is that you avoid stating explicit causal research question when you start this study, because we have all been trained to avoid using the word causal. And then people estimate associations using regressions. They can even say that we remember that causation is not, that association is not causation, and then they rely on these methods to justify interventions. And in medical literature, it's exceptionally common, with over 90% of publications, according to some meta-reviews, actually have deep misalignments between goals, methods, and interpretations. And one of the deeper problems is not even thin expertise in causal inference, but oftentimes lack of awareness that we actually even need that expertise, because when we lack awareness of a problem, we cannot solve it on the governance level. So these have been some of the examples of the impact of hidden assumptions. This is probably less than 1% of the hidden assumptions we make routinely in drug development. And just to leave you with the core idea, this is the core idea. This is the core illustration that I hope will stay with you, and I will try to write more on this topic on my LinkedIn, so if you're interested, check it out. Thank you.
Thank you, Dr. Tokavitsky. And I think we are running a little bit late, but we still want to take one or two questions from our audience. So, any questions? So, yeah, can you hand the microphone?
I have a question for the second speaker, Dr. Colonel.